Yeah.
Yep.
Yep.
Looks nice. Yeah.
All right. So welcome everybody. I am Marleen de Bruijne and I lead the model-based medical image analysis group at the BIGR. And today I want to show you some of the examples of the type of work that we do and also the kind of master projects that you could do in our group. There are a couple of specific projects proposed and you can talk about those in the chat room later today. But if I show something else that you think is particularly interesting, then also feel free to contact me or one of the other group members about possible other opportunities. This is the group, a great team of people having lots of collaborations and having weekly research and brainstorm meetings online that you will join if you are a master thesis student in our group. Our group focuses on machine learning techniques. We develop and validate such techniques for quantitative analysis of medical image data. And the final aim is to make large-scale clinical research easier and to help clinicians make better, more personalized decisions. We work on various applications. Brains, lungs, vessels are the main ones. And we focus on fully automated analysis, large-scale studies, and extracting imaging biomarkers that tell you something important about the risk of developing specific diseases. Machine learning, and then especially deep learning, you've already seen lots of examples today, is a very hot topic in medical imaging research. And these techniques work really, really well on very specific, very well-defined problems. If there are sufficiently many carefully annotated examples, typically manually annotated, which takes a long time. And when the target data to analyze is similar to the training data that we trained our models on. And often that is not the case in practice. So a lot of our research in our group focuses on how we can learn effective models in real life situations. One of the examples of the things that we're looking at is whether we can directly predict disease based on the image. So a more traditional approach to medical image analysis would be if there's an input image coming in. For instance, here you see a multimodal MRI of the carotid artery, the artery in the neck. We would perform a segmentation, in this case, of the plaque and the different plaque components, which tell you something about the risk that the patient has that this plaque will rupture and then finally cause a stroke. And what we want to do instead of relying on these manual annotations and this knowledge that the radiologist has on what factors might be important, but it might not be the only factors that are important, we want to train models that directly, based on these images, but also on other clinical parameters, predict the stroke risk. One example of such direct prediction, not yet exactly disease prediction, but prediction of white matter hyperintensity volume. This was work by a master student who just graduated last month. He showed that if we train a model to predict the volume of white matter hyperintensities, and those are these, if you see my mouse, these white blobs in the brain MRI scan. If you train a model to only predict those volumes and compare that with a model that first segments all the pixels that belong to a white matter hyperintensity and then compute the volume, the direct prediction is at least as good, actually slightly better than the indirect prediction going via the segmentation. So that's a very interesting new result that we hope to publish and was made by a master student recently. Another approach is learning from less labels, self-supervised learning. In self-supervised learning, we teach the model to solve other tasks that do not require these manual labels, but instead the labels are something you can define easily based on the image itself. But it does require a certain understanding of the image to be able to solve those tasks. For instance, it could be predicting from a slice in the image that you see, predicting in which part of the body, which anatomy it is, or which slice in a 3D volume it is. Or you could make a puzzle out of the three-dimensional image by shuffling blocks and rotating them, and then the model should learn to put them back in the correct order. For that, you need to be able to understand the model. The model will learn already what are important characteristics of this medical image, and you can use such a model to initialize or regularize your final disease prediction or segmentation model. There are a couple of example projects proposed today by Shuai Chen, our chairman, and Deep Kayal. So talk to them if you find this interesting. Another an assumption that we typically make in machine learning is that the training data that we have, that we trained our model on, is similar to the target data that we want to analyze. And if that is not the case, and it often is not the case if in practice in medical imaging, if data come, for instance, from slightly different scanners. Here you see an example of brain MRI that are similar to a radiologist, but they're made on different scanners. And to me, they look quite dissimilar. And to a machine learning model that is not trained to understand these differences, it will also look quite different. So a model trained on one of these images will probably not work well on another of these types of images. And we're looking into new machine learning techniques that are less affected by such changes in distribution, domain adaptation, that is called. We work on multiple clinical applications. An important one is brain image analysis, where we focus on early diagnosis of dementia by segmenting different tissues and structures in the brain, detecting and quantifying lesions and quantifying cortical structures. Here's one example, which was also a master thesis project in our group by Kim van Wijnen. The task here was to detect and quantify perivascular spaces, which are very small, bright lesions in these brain MRI scans. And that is also one of the topics proposed today. We want to take the approach that Kim developed a while ago as a master's thesis student in our group. She's now a PhD student supervising new master students. And we want to take the two-dimensional approach that she made and extend it to be able to analyze full brain scans, full three-dimensional brain scans. And we have lots of data available to train models for that. Then other applications that we're looking into, but I don't have any time to explain the details of this, is vascular image analysis, analyzing risk of stroke or risk of comorbidities, COPD exacerbations, and applications in lung, mostly lung CT analysis, also lung MRI analysis in diseases, COPD, cystic fibrosis, and of course, recently, COVID-19. That's all. Thank you for your attention. And if you're interested, feel free to contact me or any of the other people in the group.